Hey guys, good morning. So, welcome to my first ever video of Biolearning. I am super excited to do this. The thing is, when I was studying, I'm a biology graduate. So, when I was studying, it was always you got to mug this up and you got to put it out on that paper. That's not really what learning is about. Learning is finding something interesting. You want to know more about it. That's why we learn. It's not because we need to pass an exam, right? And the thing with biology is it's so interesting. It's so applicable to everything in our lives that knowing it is super cool. Okay? Like biologists really can do a lot, right? From genetically engineering whatever's there on the earth right now compared to solving disease, finding solutions for diseases, and you get the point. There's like so much you can do. The thing is, in schools and colleges, there's a limited amount of time, there's a lot of portion to complete, a lot of syllabus, so we generally tend to rush through it, just skim over the top, learn exactly only what we need to know to clear the exam, right? So what what my what my idea is basically with this bio learning is to give you a little of background on details about certain aspects of biology that are really pretty cool in the sense that in the end when you learn a little bit of a little bit of b, a little bit of c, a little bit of d, you will be able to mash it up and see how a actually leads to b, leads to c, leads to d. You get the point. You're able to connect, you're able to link everything, and biology is basically that. It's basically linking. I mean, for example, let's take you, a human being. Your heart is beating, right? Why is your heart beating? Your heart is beating because of the muscle contractions in the heart muscles. Why are the muscles contracting? Because of chemical reactions. You see my point? You can learn all of this individually, but unless you put it together, you don't really know what to do with it. Then it becomes all book knowledge and you don't really know how to apply it. When I was teaching, I told my students, I was teaching microbiology, so I was telling my students, you can get 99% in your exam papers. That's fine, well and good. But at the end, when you go to the real world and you have to work in an industry, do you have the knowledge? Like you can have had 99% in your exam papers, but when you reach the industry and they ask you, okay, tell me a bacteria which would eat sewage, if you don't know that, or if they don't have a bacteria, how will you isolate that bacteria? How would you use it? Applicable knowledge is the most important. Okay, so I think I've spoken too much. Let's start. Today's topic, what I plan on taking is basically the central dogma, followed by the cell cycle. These are all like really complicated terms, but they're the basics, what we have to learn the first year that you ever do some graduation in biology. Okay, look at this picture over here. Okay, you've got this whole thing over here. Complicated, right? So what is the central dogma? It's very simple. All our cells, every single cell in your body is comprised of DNA. Am I right? Right? So every cell is comprised of DNA. Now, what's the use of DNA? Everyone knows that DNA is the building block of life. They give the signs, they give the orders. DNA is like the boss of your body. Without DNA, I mean, there's no life. DNA is the reason we are alive. So from DNA, you've got like a strand of DNA in your cell. This DNA basically it's transformed to a strand of RNA, right? Now this so this process of converting DNA to RNA is basically called transcription. Now once you've got your DNA to be converted to RNA, this RNA, not when you hear something about muscle contractions or when you hear anything happening in your body, it's not the RNA that's doing that work, it's the proteins. It's the proteins. This process of turning the RNA into proteins is called translation. Yeah, I'm sure you've heard of enzymes. What enzymes are? Enzymes are proteins, right? Enzymes are the reason that a lot of chemical reactions take place in the human body and in the world. Enzymes are proteins. Basically, every single thing that happens in a living system is the result of proteins. There is nothing that can happen without proteins. So, um, now what is really, really cool in the human DNA? I'm just talking about humans in this lecture. What is really cool in the human DNA is you've got a cell, right? You've got a cell and you've got DNA inside. I'm just drawing the random DNA. Okay, just assume that's the DNA. It's a really bad drawing, but in any case. Now, give me an example. Have you heard of fingerprinting at crime scenes or for paternity tests? They want to compare the DNA of the father and the child for paternity tests, or a criminal with a sample they found on the crime scene, for example. Is there a particular part on the body that they have to take a cell from to get that particular DNA? No, there's not, because now think about this very carefully. It's really cool. All the cells, each and every one of your cells in your entire body has the exact same composition of DNA, which means whether I take a cell from your skin, or if I take a piece of your hair, or if I take from any different part of your body, I get the exact same DNA. Now pay attention. I told you DNA is responsible for giving you RNA, which is responsible for giving you proteins, which is finally responsible for what happens in that cell, how that cell behaves. Proteins are responsible for how that cell behaves. So you tell me, when you have a person, when you have a person, why is it that the cells in the person's hair are different compared to the cells in the person's arm, compared to the cells in the person's stomach, compared to the cells in the person's eye? All these cells are different. They don't all look the same. Hair can be a different color. Eye has got some gelatinous substance. The stomach secretes acid to digest the food. The hand has got skin and blood vessels. You get my point. They're so different. So how is it that if all the cells in the human body have the exact same DNA, each of them ends up looking and behaving differently? I'll tell you. So you've got a cell in the hair, okay, or let's say head, and then you've got cells in the arm. Now I've got this much of DNA in both. All the DNA will not be converted into proteins. This is a very, very important part, important concept to get. All the DNA cannot be converted into proteins. Okay, in the hair, in the hair, only this part of your DNA will become proteins. Okay, only that part. Whereas when you come to your hand or your skin, this part of the DNA is responsible for the proteins available in your skin. You get my point? See how different proteins are made in your hair and your skin. Okay, in your hair you've got a different part of DNA, and your protein you've got a different part of DNA. This is so cool. This is why you've got this exact same DNA in every single cell of your entire body, and yet you've got so many different types of cells in your body. Okay, that's basically the central dogma. Now, coming to the packaging of DNA, this is basically what all of us study in what DNA looks like in our graduation of biology. Even in the 11th, 12th standard of school, we study that DNA looks like this, right? A right-handed cell. We've got the helical structure with nitrogenous bases in between. Now, This DNA, do you have any idea how much DNA you have in a human cell? A single cell? It's a lot. Like it's a lot. Now, can you imagine the size of a cell? Okay, just look at your hand. Can you see the cells? No, you cannot. They're so small. So, imagine how much DNA has to be packed into that one cell. And I told you that DNA has to have enough compartments or enough pieces to secrete enough different types of proteins to be able to give an entire human body. So all that DNA, if you just scrunch it up and sort of push it into that cell, it's not going to fit. It's definitely not going to fit. So this DNA is packed into this. Basically, what happens with this helical structure that you have is it is rolled up in itself. Okay, it's rolled up. Then this is rolled up again. Then this is rolled up again. It keeps rolling into itself again and again and again until you get a structure like this, which is called a chromosome. Now, the entire packaging system of DNA is much, much more complicated than what I've just drawn here. So don't take this as final. I will put another video with packaging of DNA. But basically, you get a chromosome. Now, the thing is, we are always shown DNA in this X-shaped structure, right? When DNA is in the cell, it's not exactly permanently in the X-shaped structure. This X-shaped structure comes into being only when the DNA is about to divide. Let's do this. Now, the central dogma that you just learned, it's got to balance DNA out, right? Now, in a human cell, we know that we have 46 chromosomes and 23 pairs. Now, what does that mean? We've got a cell, right? We've got a cell. We've got one chromosome, another chromosome. Notice I am not drawing an X. I am not drawing an X. I am drawing straight lines because in the cell, this is how a DNA looks. You've got one chromosome, another chromosome. Then you've got third chromosome, fourth chromosome. Then you've got fifth chromosome, sixth chromosome. Are you getting what I'm doing here? Are you understanding? This is one pair. This is another pair. This is your third pair. That is how, in the end, in one human cell, you have 23 pairs and 46 chromosomes. Right, a chromosome looks like this. So, what happens when this cell has to divide? I mean, the cell doesn't remain the way it is forever, right? You grow from a small baby to a huge human being. Obviously, the number of cells in your body has to keep dividing. Now, the problem is when a cell divides, it should not lose any of its DNA. Okay? That's the point. This cell undergoes something called replication. Replication is what happens before transcription and translation, which is what we saw in the previous slide. Replication is only multiplying or copying of the DNA strands. Now, when you undergo replication, you get the same cell. The difference is one chromosome is replicated. This is one chromosome replicated to give you something like this. This is the X structure we all know so well, right? Same way, one chromosome is replicated to give you this. Can you relate this image with this image? We've got one chromosome here, one chromosome here. This is a pair of chromosomes. This pair turns into this. These are your pairs of chromosomes in the first cell. Basically, what happens, one chromosome divides, sorry, not it replicates, it gets a copy. This is called a sister chromatids because they are joined in the middle. Can you see them joined in the middle? These are called sister chromatids. So, when this cell divides, each sister chromatid is, let me just change the color here, it's becoming too complicated. Each sister chromatid is pulled apart. Each sister chromatid. See, these pairs are not pulled apart. It's the sister chromatids after replication which are pulled apart. So, in the end, when you get your new cell, I should have drawn this in a different color. Okay, imagine the red ones are your original DNA and then these green ones are your sister chromatids. Right. So, in the end, you will end up getting something like this. If you can understand by the colors and the drawings, each sister chromatid is pulled apart. So you end up getting the exact same composition of DNA as you saw in the first slide. You end up getting the exact same DNA composition as you saw in your parent cell. This becomes your parent cell, and these are your daughters. Don't ask me why they're called daughters and not called sons, but they're called daughters. Anyway, okay, I hope you understood this concept. Then we go to the next step. Have you heard of something called meiosis? What we've done till now is called mitosis, which is the basic replication of DNA. Mitosis. Now we come to something called meiosis. You see, in certain cases, like for example, when we have gametes of a human being, for example, the sperm and the ovum, you have to divide the total concentration of DNA from the parent cell so that ultimately from the mother and the father you have to get half your genes from the mother and half your genes from the father. You cannot take a full cell from the mother and a full cell from the father. You get a very odd looking child. You have to take half the cells from the mother and half the cells from the father. So, in that case, how can we convert this cell, which is a full diploid cell, how do we convert it into half a cell? By that is done by the process of meiosis. Okay, let me draw that cell again. So I've just got you a picture to show you what meiosis is. Remember, this is your parent cell, what we saw in the previous slide. Yeah, see, parent cell with pairs of chromosomes are shown right here. After replication or mitosis, you get, sorry, you get sister chromatids. Now notice that this is one pair and this is one pair in the parent cell, right? So the two members of this pair which are shown here are called homologous chromosomes. During meiosis, the first thing that can happen is the homologous chromosomes are separated, not the sister chromatids as in meiosis. The homologous chromosomes are separated during meiosis 1, which will give you still diploid cells, which we don't want. We want haploid cells for meiosis, right? So the second meiosis, meiosis 2, you have the sister chromosomes which are pulled apart to give you 4 haploid cells. These are what form your gametes. Got it? The actual process of meiosis obviously is much more complicated. This is just the basics of what happens in meiosis. Coming to the cell cycle, how does all of this relate to the cell cycle? You see this M over here? This M stands for mitosis. The cell cycle basically comprises of 4 parts. One of those is mitosis. Now we've already seen what happens in mitosis. After mitosis, where you've got 2 daughter cells, 2 diploid daughter cells, these these go through the G1 phase. Now in the G1 phase, basically what happens is these daughter cells, after they're just born, they're very small. They kind of need to put a lot of time and effort into making their own new proteins or whatever it is that they need from their own new DNA now. They need to basically grow in size. These daughter cells need to grow in size. They need to make proteins, they need to make ribosomes, they need to make whatever organelles that they need for a fully functional cell. So this entire G1 phase, G stands for gap. This entire G1 phase is taken to make the daughter cell grow large enough so that it is the same size as the original parent cell. Imagine this parent cell dividing to give you 2 daughter cells. Obviously, the daughter cells are going to be smaller in size, right? So this daughter cell takes the entire G1 phase to become the same size as the parent cell. That is one point. The second thing that happens in the G1 phase is you've got your DNA, right? This DNA needs to be replicated again because it needs to be replicated to undergo back to mitosis, right? So in the G1 phase, the daughter cells calls upon certain protein molecules which are required for replication. Replication is again a very specific process. You cannot afford to make mistakes because the DNA has to be identical. Otherwise, suddenly in one part of in your hand, you'll suddenly get a boil, or you'll get maybe a different color patch of skin, or you'll get some abnormality, right? Which doesn't happen all that often, which means replication is a very strict process. So it collects in the G1 phase, the daughter cell collects all those proteins that are required for replication. S, coming to the S phase, stands for synthesis phase. This synthesis phase is the phase during which replication occurs. This entire synthesis phase is the phase during which replication occurs. That means the 2 homologous chromosome pair turns into what you see here, with each chromosome having a sister chromatid. Got it? This is the purpose of replication. After synthesis is over, after you get this, you go into the G2 phase, which is your gap 2 phase, wherein they check that all the chromosomes have been replicated, that the cell has increased in size, that it is ready to go to mitosis. G2 phase is basically a checkpoint. They've got checkpoints to check whether the cell is ready to undergo mitosis. After the G2 phase crosses its checkpoint and it is sure that the cell is ready, it will finally allow the cell to undergo mitosis. Now, the actual process of mitosis is not simply replication. You've got replication, then you've got um, fibers which will pull the sister chromatids apart. Then you've got one cell dividing, see the division into two daughter cells. The entire process is much longer. It's got a lot of proteins involved, it's got a lot of fibers involved, it's got a lot of things involved. All that happens in this small little region called mitosis. And ultimately, you end up getting two daughter cells. Again, the cycle starts again. That is the normal cell cycle. Now, you have only gametic cells, this gametic cells which have to become haploid. That is your sperm and ovum. Only these cells will undergo will leave the normal cell cycle and undergo meiosis. See, the normal cell cycle for all cells in your entire body includes G1, S, G2, and M. Only the gametic cells, that is your sperm and ovum, will leave the cell cycle to undergo meiosis. That is, when it reaches, it starts here. When it ends with the G2 phase, that is when your cell has replicated and it's got chromosomes with sister chromatids which look like this, it is ready to either undergo meiosis or mitosis. Now, do not forget, mitosis is for every single cell in your body except your gametic cell. If it is a gametic cell, it will undergo meiosis, which will end up giving you four haploid cells. I'm just drawing one, but you can imagine how many pairs there are. So that's basically what I had included for this video, the point of mitosis, meiosis, replication, your whole arrangement of cells, the whole arrangement of chromosomes, how they are segregated into your daughter cells. I hope this video helped you. If you like this video and you want more, please like and subscribe. Like I said, this is my first ever video, so apologies for um, problems. I mean, whatever, maybe if I could improve myself, please, by all means, please comment. I would love to hear from you. Yeah, thank you so much for listening. I hope this helps you. Thank you.